to BioSpectrum's news bulletin. Headlines for today are: IISc researchers develop a novel method for delivering aspirin. Johns Hopkins scientists develop a nanochemoparticle to treat bladder cancer. Researchers in China develop a stretchy fiber to capture body motion. Moving on to the main news now. IISc researchers develop a novel method for delivering aspirin. An interdisciplinary team of researchers at Indian Institute of Science in Bangalore has come up with a novel chemical compound made of a sugar molecule called xylitol that can be tailored to release aspirin, which is known to reduce fever and pain over a desired duration in the body. This unique drug delivery system that can carry a large amount of drug is not only harmless to the living cells but can also be easily injected. The researchers modified the reaction conditions in such a way that xylitol was forced to form a long straight chain that can be easily dissolved into a gel and then injected. The linear polymerization was achieved using a novel technique. The technique can also be used to develop similar delivery vehicles for other drugs with sustained and tunable release. Johns Hopkins scientists developed a nanochemoparticle to treat bladder cancer. Johns Hopkins researchers have developed a way to successfully deliver nano-sized platinum-based chemotherapy drugs to treat a form of bladder cancer called non-muscle invasive that is found in the lining of the organ and has not invaded deeper into the bladder tissue. The tiny drug-infused particles potentially offer a less toxic clinical alternative to standard chemotherapy delivered intravenously or through a catheter inserted into the bladder. The team of investigators engineered the nanoparticles from biologically compatible materials such as polyaspartic acid and coordinated them with molecules of the chemotherapy drug cisplatin, considered a first-line treatment for human bladder cancers that have spread through the organ's inner lining into its muscle tissue. Number of rats which were treated with the drug nanoparticles showed exciting results. That is, no rats treated with the drug nanoparticles had signs of high-grade tumors 16 weeks after the treatment. The team hopes to initiate early-phase clinical trials to evaluate the safety and efficacy of this treatment for patients. China researchers developed stretchy fiber to capture human body motion. A team of researchers at Tsinghua University in Beijing offers the first demonstration of optical fibers sturdy enough to sense a wide range of human motion. The new fiber is sensitive and flexible enough that it can detect joint movements unlike currently used fiber sensors. The researchers have a mixed fluorescent dye called rhodamine B and they have mixed this into silicon. When light shines through the fiber, some of the light is absorbed by the dye, and the more the fiber stretches, the more light the dye absorbs. So simply measuring the transmitted light with a spectroscope provides a measurement of how much the fiber is being stretched or bent, which tells an observer about the movement of any body part it is attached to. Thank you.